And now I have the great pleasure to introduce Professor Stanley. Margaret Stanley will address the immunological issues of infections and disease. And this is a very important part in where immune response and in what we see and what we've been discussing since the morning. Margaret, please.
This is going to be a very simple talk. Immunology can be very complicated, but it has some really basic principles. So I've entitled this talk, The Immune Response to Natural Infection, or How to Avoid the Forces of Law and Order. And the forces of law and order are, of course, the defense forces, immunity. And immunity we now think of as innate, or natural immunity. It used to be referred to as non-specific. That's not true. It's actually got specificity. We used to say it had no memory. That's not true. There is actually some innate memory. But the second arm of the immune system, which is adaptive or acquired, there is no doubt that is antigen-specific, and it has an exquisite memory for the antigen it sees. Now, innate and adaptive immune systems are indeed a partnership. I always say they're like a good marriage. And a good marriage means there's a lot of give and take and excellent communication. And that's essential in generating a holistic defense system which counteracts pathogens. And the innate system has pattern recognition receptors, PRRs, and these PRRs are actually there to sense danger. They sense when cells are injured. They sense when cells are stressed. They sense when cell death exceeds a threshold, because you can't rush in with your defenses unless you have something real to fight. And when the pattern recognition receptors sense these, they activate a whole cascade of events, some of which are killing events, attempting to restrict the pathogen, to remove it by removing infected cells. But others are communication. They're communicating to the cells of the adaptive system so that you actually generate an immune response, an adaptive immune response, which is appropriate for the infection you have. Now, I wanted to look a little bit at these pattern recognition receptors. About 5 years ago, I could give a very simple talk on these because we really only knew about toll-like receptors in detail. Now, it's very complicated. There are receptors in the plasma membrane, and you can see the TLRs, toll-like receptors, TLR1, 2, 6, 5, and 4. There are other receptors which are not TLRs, but which sense molecules in pathogen coats, and these are the dectins. And then when you go into the interior of the cell, in the endosome, there are another set of TLRs on the endosomal membrane. And then there's a whole set of receptors which are in the cytoplasm. Now, how you have to think of this is rather like playing those computer games where you've got the hero trying to get to a target, and that every time the hero moves, there's a defense system pops up which he has to avoid or remove. And then there's another one pops up which he has to avoid or remove. And this is exactly what the innate immune system is about. So the TLRs recognize molecules, but the ones on the plasma membrane recognize molecules that are inherent in the coats of pathogens, lipopeptides, flagellin in bacteria, lipopolysaccharide. The dectins also recognize molecules in coats. When you go to the TLRs and the endosome, they are recognizing nucleic acids, which are in an inappropriate place or an inappropriate structure. And you see that in the cytoplasm, you also have a set of receptors recognizing nucleic acids. Now, you'll see there's the once the receptor recognizes its ligand, it sets out a signaling cascade which activates transcription factors, which are global transcription factors. They actually switch on a whole set of genes, and these genes encode, in most cases, important cytokines, pro-inflammatory cytokines, or they encode enzymes or proteins which will target pathogen fragments or pathogen viral particles and target them to the proteasome for destruction. I'm spending a lot of time on this because this is an absolutely crucial part of our defense system. You've got to remember that 90% of all infections are actually handled by the innate immune system without ever recruiting the adaptive immune system to it. So these are important. And finally, I want to point out that one of the things you start activating are the cascades, which cause the cell to die. So innate immunity is about setting up killing systems, but it's also about communicating the nature of the death. And there's a lot of crosstalk. Everything is talking to everything else. It's an integrated defense system. So let's go back to this innate and adaptive systems. The innate system we think of in cellular terms as phagocytes, innate lymphocytes such as natural killer cells. We think of soluble proteins such as complement. Importantly, you've got to think of epithelial barriers. Remember, this is a virus that only lives in the keratinocyte, and the keratinocyte is a very key player. It's not standing by, it's there participating fully in the innate immune response. Now, the cytokines and molecules that are generated by these pattern recognition receptor ligand responses kickstart the adaptive system. And again, we think simply of this as in two arms. Humoral immunity, which is antibody-mediated. The function is to neutralize extracellular pathogens to prevent infection. The generals of that system are CD4 T cells, and they bear the surface marker CD4. And in humoral immunity, we call these generals Th2. If you have an infection, you've got to have a killing response. And so you have cell-mediated immunity, Th1 cells, CD4 positive T cells, and their function is to arm cytotoxic effector cells to kill infected cells. So just again, the adaptive immune response, how the generals are working. The Th1 cell-mediated immunity, you've got CD4 positive T cells that secrete the cytokine interferon gamma. The job is to generate a whole cytotoxic killing set. The cytotoxic antigen-specific T cells bear the surface marker CD8. You also have activated macrophages. The cytokines themselves are killers. You also have innate lymphocytes, such as natural killer cells. But the whole purpose is to kill. And then Th2 cells, here you're secreting cytokines that help B lymphocytes make antibody to prevent reinfection. Now, unfortunately, T cells are much more complicated than Th1 and Th2. And here in a very simple way, I want to show you some of the subsets of CD4 positive T cells. Now, they all have different functions in the defense response. So we have the Th2 subset, who are going to generate differentiating B lymphocytes to make antibody. Then you have the killers, the Th1 subset, which will interact with to help CD8 positive cells kill, to help natural killer cells. And then you have another set, Th17. But essentially, these are killers. These are the warriors. And then you have these guys at the bottom, which we call regulatory T cells. They are the diplomats. They're there to control and calm down the situation. So they turn off the killers. So, and that's very necessary. Once your army has been successful, you want it to go back to barracks and not do any further damage. And that's what the function of Tregs are. Okay, so how does all this fit into a natural infection? Now many of you will have seen this cartoon innumerable times, but it still serves its purpose. Because you've got to remember that HPV infection takes a long time. So you get infected, then you have weeks or months where in the infected area you can't actually detect DNA. Then lesions appear for reasons we don't quite understand. And again, those lesions persist for weeks or months. And eventually, there's an immune response. And as John commented, this is a cell-mediated immune response. But what triggers it is still not clear. At the same time, you probably have seroconversion with antibody to the L1 protein. And then, as we know, most people, particularly in the cervix, will get rid of their DNA, they will have a sustained clinical remission, and they probably have latent infections, and that will be discussed later by Patty Gravett. But a proportion of the population do not make a good cell-mediated response. That's the key thing. They stay DNA positive, the virus persists. These are the people who are at risk for going on to high-grade disease. And all of this, because it's taken a long time to tell you about it, takes a long time to happen. So let's have a look at the cell-mediated immune response. I'm just going to summarize this, because one of the things that's crucial is to recognize that cell-mediated immune responses to HPV are local. Local responses are the key here. And that's defeated investigators for over a long period of time, because immunologists classically take blood, separate out white cells, and try and look for antigen-specific responses. Now, those systemic responses to HPV antigens are extremely weak. And so you're right at the edge of the sensitivity of your assay methods. And it's one of the reasons why the field has not advanced as fast as we would like. What we do know from the work of Sjoerd van den Berg and his colleagues is that you have memory to the E2 proteins and the E6 proteins in people who apparently have successfully cleared infections. Some work that was done by my colleague Yinling Wu from Malaysia in a prospective study showed that women whose CIN1s regressed had CD4 antigen-specific HPV16 E2 and E6 responses, but the E2 responses dominated. What we also know from Yinling's studies was that the regression of CIN1 correlated with the presence of functional cytotoxic lymphocytes. Lymphocytes that had the capacity to kill were present in the epithelium of CIN1 lesions, which then regressed. But the key thing is that this is a local response. And if you look when genital warts response, there is a massive infiltration of mononuclear cells, but systemically, you struggle to actually show the presence of these. Everything is happening in the local lymph nodes and in the local draining systems. So these are the questions which I'm going to briefly respond to. Why does it take so long? How does it avoid host defenses? Why do they persist? Why do the tumors arise despite these defenses? And very briefly, why does vaccination work so effectively? John has gone through this, but I want to reiterate, this is a completely intraepithelial infection. There isn't any viremia. It takes a long time, and there is a very controlled viral gene expression. And there may be different defense responses at different times as the virus goes up through the epithelium. So that intraepithelial life cycle is in itself an immune evasion mechanism. First of all, there's no viremia, so the systemic defenses aren't activated rapidly. There's no cell death. Very clever virus. This doesn't have to kill anything. It's making virus particles in cells which are already destined for death, so there's absolutely no reason to kill it. Because there's no death, there's no inflammation. Now, in view of what I told you about those in pattern recognition receptors, you would ask the question, hey, you've got a cell packed with viral DNA, viral proteins are being expressed. Why don't all these receptors get activated and turned on? What's happened to innate immunity? Where are the antiviral cytokines? And that's the great secret of this virus. Essentially, it makes very little protein in these dividing proliferative compartment of the epithelium. There isn't a viremia, so you don't have systemic activation. No cellular death, no inflammation. Crucially, it globally downregulates pattern recognition receptors in the proliferating cells of the epithelium. And the consequences of this are huge. So E6 and E7 downregulate the type 1 interferon response. Interferons are crucial antiviral defenses. This virus absolutely suppresses them. E6 and E7 downregulate toll-like receptors. They downregulate TLR3, and that is work from my colleague Nick Coleman and Mark Pett and their PhD student Emily Barker. We know that E2 downregulates one of the cytoplasmic receptors, RIG-I. And both of these things are important because they prevent the secretion of pro-inflammatory cytokines, which would then switch on innate adaptive molecules such as the Langerhans cell. All that is being blocked because there's no signal coming from the innate system to activate adaptive immunity. Langerhans cell maturation blocked by L2, and Langerhans cell migration blocked by E7. So you don't have any activation, and essentially you're evading the innate response and delaying the activation of adaptive immunity. It's a very high-risk strategy, and it's high risk because it prolongs the replication of HPV in the epithelium. So in low-grade disease, you are evading innate immunity, you're delaying and activating cell-mediated immunity. That allows for the accidents that lead to deregulation of viral gene expression and the production of an immunosuppressive milieu, which actually prevents any systems recognizing the transformed cells. Just very briefly about antibody. What about the role of antibody? Well, as you know, you make antibody responses to L1, and the neutralizing epitopes, neutralizing antibodies rather, recognized conformational epitopes, those that are in 3 dimensions. If you look at the antibody response in natural infection, frankly, it's wimpish. You make a neutralizing antibody, it's to L1. Mainly type specific, it's only 6 and 11 that have cross-neutralizing antibodies. The response is incredibly slow. It takes, on average for HPV 16, 9 months for seroconversion from the first detection of DNA to the actual appearance of antibody in the serum. Not everybody seroconverts, but the antibodies do persist, and this has been shown in at least 2 studies. And the antibody generated in natural infections is protective in a large number of cases. So why, if you're looking at that, you wouldn't have thought that a vaccine that generated antibody would work. So why do the VLP vaccines work so well? Because they do. But everybody seroconverts. Everybody. Nobody doesn't make a response. They make loads of antibody. The antibody lasts. But we still don't have an antibody concentration that correlates with protection. And you would then say, well, does the antibody actually do something? And the old experiments in dogs and rabbits showed absolutely that when you did the classic passive transfer of antibody, you could protect naive animals. And I have to say that's the only concrete evidence we have that the antibody generated by vaccines has a protective role. So why are these responses so much stronger? Well, it's the route of immunization. Natural infection, you can it's hard to get virus to lymph nodes because you don't have a viremia. You're shedding from the mucosal surface. You don't get to a lymph node, you will not activate adaptive immunity. The vaccines ignore the epithelium, they go straight into the muscle, so you have very rapid access to vessels. You have an absolutely perfect immunogen, and you get an excellent response. And you get all the features you want of a strong response. And you get very good memory, both in terms of persistence of antibody and the recall response. So in a natural infection, you've got these very common infections, most of them resolve, resolutions due to cell-mediated immunity, in most cases. But in men, I will speculate now that data which Anna Giuliano talked about this morning, where you get very little antibody in men, I think most of the infections in men are cleared by innate immunity, not by adaptive immunity. And the keratinized surface of the penile shaft is a very good barrier. Now, neoplastic progression is associated with immunosuppression and correlates with a reduction of cytotoxic killers in the epithelium. And then antibody levels in natural infections are low because there's no viremia. And seropositivity does give some protection, but it's not complete, in contrast to the vaccines, which give you excellent protection. So I'll stop there. Thank you very much.